phosphorylation. Probably thinking, why have I got all this writing on the paper already? Because I'd normally write it as I go. The answer to that is because my webcam didn't work properly, so I apologise for that. So you've got all the notes up there already. Right, I'm going to discuss them with you now. Okay, so oxidative phosphorylation takes place in the crystal, which is this inner membrane of this mitochondria. Mitochondria, you need to know the size of it. So it's 2 to 4 micrometers in length, and 0.5 to 1 micrometer in diameter. Because if you remember, you should remember that they can vary in size. They're not all exactly the same size, these mitochondrial organelle in cells. Some are bigger and some are smaller. If you remember from our variation video that things can be different sizes for various reasons, but anyway, I'm not going to discuss too much about that with you now. So in your crystal, what basically happens is your FAD and your NAD actually provide these hydrogen ions, from the hydrogen atoms, which split into the hydrogen ions and electrons. They provide that, and the hydrogen ions basically flow across the crystal, this inner membrane, into the intermembrane space. Now you're probably wondering, why do they do this? Well, it starts off because the hydrogen atoms release these hydrogen ions. All of a sudden, the potential or the electrochemical gradient forms between this side of the membrane and this side of the membrane. So it flows from the matrix up here into your intermembrane space, which is down here. We haven't discussed this in too much detail before. This bit here, the crystal membrane, this consists of hydrophilic heads and hydrophobic tails. Now, hydrophilic just means water loving, and the hydrophilic heads basically like water, so they point outwards, because obviously the cytoplasm is a jelly-like substance and it has moisture in it. And the hydrophilic and the hydrophobic tails, hydrophobic phobia or something means you don't like it, hate water, so the hydrophobic tails are on the inside of the membrane. In the membrane, there's also these protein carrier channels, which are where these H+ ions actually flow through in order to reach the intermembrane space. As the potential builds up here, it flows through these protein carrier channels, and the energy provided from this is used to transport electrons, which is used to basically transport electrons, and the electrons bind to molecular oxygen in order to create your water molecule. Because basically these H atoms, from the FAD and the reduced NAD, the reduced NAD gives up its H atoms, which then split into ions, electrons, and oxygen, which comes from just the area around in general. And what these do is these are used to produce ATP again. So basically this whole process is used to make some more ATP. So what happens is your H+ ions move across this protein carrier in order to reach the intermembrane space. So once they're there, this ion concentration builds up, which basically means that these H+ ions want to get back to where they started off, which is in your matrix. So you're probably thinking, well, how do they do this? Well, this is via a channel which is associated with an ATP synthase enzyme. ATP synthase, clues in the name, ATP is synthesized from this particular enzyme, or this enzyme aids the synthesis via ADP and PI. You should know that from previous videos that ADP is adenine diphosphate, as opposed to adenine triphosphate. No, I said adenine, didn't I? I actually meant adenosine triphosphate and adenosine diphosphate. So adenosine diphosphate and inorganic phosphate joins together in order to form adenosine triphosphate, which is your ATP, which is used for energy. So these H+ ions provide the force for that, and they flow back into your matrix, which means the concentration in your matrix of H+ ions builds up, which also joins with these electrons that are also split up from these H atoms, because the structure of an atom has electrons, protons, and neutrons in the nucleus, and the nucleus is in the middle of the cell. So some of these protons and electrons are used in the process in order to generate water. So basically, this part of the process generates the water. If you remember, you cast your mind back to like key stage 3 and 4, you should know that in the process of respiration, generates carbon dioxide and water, and that's basically how the water's formed, and the carbon dioxide is like formed as well throughout this whole process. But the key to focus on for this bit is that the ATP synthase enzyme joins the inorganic phosphate and your ADP in order to form this. Now I've wrote some numbers here, you're probably thinking, well, what are these for? Well, basically, where I've wrote this number 1 is where step 1, or glycolysis, of aerobic respiration takes place. Steps 2 and 3 take place in the matrix, and steps 4, obviously this one, takes place like across the crystal, which is your inner membrane of your mitochondria. So this flow of H+ ions through this channel is known as the process of chemiosmosis. I don't know if you've heard of this before, but chemiosmosis is this flow, this idea of the flow generating force to make ATP. So I hope this video has been helpful, and if you've got any questions, do not hesitate to ask me on my channel. Do just like type them in on my channel, because I've probably left something out of this video that may be important to you. I've tried to include everything, so I hope you found this video helpful, and I'll see you in the next video. Many thanks.